Thank you for joining us this evening for tonight's program on healing with Dr. Bill Bingston. I'm Claire Noble, the program director for the Vail Symposium, and on behalf of Chris Sable, our executive director, Dale Mosier, our board chairman, and the entire Vail Symposium board, welcome. We're now in our 49th year of convening locally and thinking globally. Two items to be aware of before we get started. At the bottom of your screen, you'll see an option for Q&A. Please type your questions in there at any time, and we'll get to those later in the program. We'll get to as many questions as time permits. Tonight's program will run until 7 p.m. It's being recorded, and if you give us a couple of days, we'll get that posted on our website, vailsymposium.org. I'd like to take just a moment to thank some of the organizations and individuals who've helped make tonight's program possible. Our sponsors include the Town of Vail, Vail Resorts Epic Promise, the Antlers at Vail, and the Vail Daily. Jeannie and Dale Mosier have underwritten the winter season. Our virtual programs are sponsored by Alpine Bank, and the Vail Symposium is supported by a generous grant from the Frechette Family Foundation. Thank you to all of our donors. And if you'd like to be a donor, I'd invite you to visit vailsymposium.org to donate. Please make plans to join us a week from tonight on Thursday, March 6th at 6 p.m. for a geopolitical program on the future of Germany after Angela Merkel ends her 15-year tenure as Germany's chancellor. We'll be speaking with Dr. Constanze Stelzenmüller of the Brookings Institute and Dr. Karen Donfried, the president of the German Marshall Fund. That's on Thursday, March 4th. Now we turn our attention to a discussion of healing. Our distinguished guest is Dr. Bill Bingston. He's a professor of sociology with a specialization in research methods and statistics. He's president of the Society for Scientific Exploration, an international group of scientists who conduct rigorous investigations into scientific anomalies. Dr. Bingston has published in the Journal of Scientific Exploration, the Journal of Alternative and Complementary Medicine, and Explore. He's also on the editorial board of the Journal of Alternative and Complementary Medicine. Dr. Bingston has written a memoir with Sylvie Fraser about the healing experiences and research entitled The Energy Cure, Unraveling the Mystery of Hands-on Healing. If you check the chat box, I've put Dr. Bingston's website in there, and there's also a link to his book, which is available on Amazon.com. I also just want to remind our audience to please use the Q&A option at the bottom of the screen to type your questions. And Bill, I'm going to hand the program over to you.
Thank you. Appreciate the invitation. And I'm going to try to share a screen here. I'm assuming I have those rights. And am I, we gotta, sorry folks. I think we're there. Thanks. Again, thanks for the invitation. Happy to be here in this Zoom land kind of pandemic. I've changed the title a little bit. I'm still playing around with the title. But basically, the short version is, for those who don't know, I've been researching healing for, I don't know, something like 105 years. And I've moved into real practical applications of the stuff. And I'm wondering, and I'm working in independent labs about whether we can capture, store, and reproduce healing without the healer. And if we can do that, and we can turn this into a scalable system, we might even be able to make this conventional. And so that's the general idea. So you can look at my website if you'd like. Here it is. You'll see it also at the end, and it's probably on all the material that's been posted. So in my quick talk today, here's the plot. I'm going to give a very brief background to previous healing studies. I have a lot of studies, and I'm only going to do a little snippet here, but you'll get an idea about what I've been working on for all these decades. And I'm going to show you then some experiments indicating that healing can be stored, and it can be stored in both organic material and inorganic material. And it turns out it can be stored. I'm now up to number 3. It can be stored in a bunch of material. It can also be recorded. And I've done this in a variety of recording media. I'm going to show you only one here. And I'm going to show you another way. I'm up to number 4, that we can enhance healing. And I've been informing into water, tap water, healing intention, and I've been able to juice it up with stuff, with gizmos, as well as with intention. And then finally, we'll finish up with speculation and thoughts on going forward. So this is the general plot of what I'm going to try to very briefly go over in this talk. So in my 125 years of doing healing or healing research, I'm interested in the parameters of healing. And this is kind of a wide statement, a broad statement. Does distance matter, for example? That's a parameter of healing. And the answer is no. But does belief matter? And I can go down that road a little bit. Belief seems to matter a little bit. I think belief retards healing a little bit. It's not going to hurt you too much, but I wouldn't recommend belief. And so I can go into all sorts of parameters of healing. Not is it possible to heal? Way past that, is it possible to understand the mechanisms, the correlates, and all those things? And so I'm interested also in the physiology of healing. What happens to the healer? What happens to the healee? And maybe in particularly Q&A, we'll touch on that. I'm interested in, can we teach this? And I do teach at selected times, people to reproduce what we're doing here. I don't have any secrets. I don't have, I have this little secret technique that nobody can have. It's all published. The mother lode to me would be the 4th bullet down, and that is the development of a theoretical model. How do we explain this stuff? And let me just tell you now, so you don't get all worked up. I don't know. I've only been doing this for 130 years. And so I'm not close yet to developing a theoretical model. And so I decided, because I'm only going to do this another 90, 100 years, and then I'm going into semi-retirement. The last bullet, development of practical applications, is because I'm going to fail at the 4th bullet. I just don't have a high hope that I'm really going to get a deep dive into a theoretical understanding of this stuff. It's very, very, very complex, and it's a very interesting problem. But here's the deal. Even if you don't understand something, is it possible to make it work? And can you turn it into something practical? And the answer obviously is yes. So we don't need to understand gyroscopes in order to be able to ride a bicycle. We don't need to understand the physiology of walking in order to walk. We don't need to understand, you get the idea. We don't need to understand what gravity is to bring it into use. And I'm trying to turn this stuff now into something that is conventional. Engineering-wise, we can turn it into something practical. And that's really the thrust here. So as said in the introduction, sometimes I write for a general audience. The piece on the left is just, it's a memoir. The piece on the right is a training course. And so we're not going to get to details of how the method was done today. But if you want, here it is. Do it yourself. Take a workshop. Listen to the CDs. Try it. I don't care if you believe the stuff, but if you practice this, it's reasonably interesting. Most of the time, I spend writing for other academics, and so I do geeky things, and I put it in scientific journals, and nobody reads it. But it's kind of the place where academics play. And if you'd like to read some of my publications, I think on the bengstonresearch.com site, Margaret does this all. I think there's 30 or 35 papers. You can get an idea if you're interested in the geeky side of all of this stuff. So I'm going to talk about a couple of experiments with mice. And I'm going to show you kind of a very simple, straightforward mouse model and give you an idea that I've done mice research quite a bit in quite a number of places. So these are cancer experiments, because I've done most of my work on cancer. These are cancer experiments on mice. And so you'll see a mammary cancer City University of New York, 2 experiments at St. Joseph's College, 1 at Arizona. You get an idea. I've done a lot of these things. Sarcomas at University of Connecticut Medical School. I've done oncogenic mice. We've been studying nude mice. We've been studying extremely aggressive mice. And at this point, I have something like 18 mice experiments, and one's actually just begun. Before I came here, I just came from a lab meeting, and I'm going to have 2 more going on. The research is ongoing. I'm not talking about stuff here that, way back in the good old days we did. It's ongoing now. I got 3 labs going. So here's the general idea. And this is the essence of the plot boiled down to a couple of pictures. I ask very simple questions. So I take, for example, a mouse model, and this is one. I take a mouse model that's already very well understood. It's been studied ad nauseum. In this particular mouse model, there's something like 2,000 published papers on it. I mean, this is literally true. So there's something like 2,000 published papers on it. Everybody knows about this. Everybody knows what goes on, meaning everybody in oncology. And the short version here is this. So we're not going geeky. We get a particular kind of mouse, and we get a particular kind of cancer, and they're matched. And we give the mouse cancer, and then we see what happens. In this particular mouse model, all mice, without exception, die within 27 days. We'll call it a month. Call it February. So they all die in a month. There's never been an exception. All mice all over the world playing with this, and it's studied all over the world, playing with this, they all die within a month. And then researchers do stuff. They try to figure out a therapy for it. They try to radiate it. They try chemotherapy. They try nutrition. They try immunotherapy. They try whatever they can try, seeing if you can make it more than a month. And so far as I know, I'm the only person who's made it more than a month. So I interject a single variable into a system that's already thought to be known, and I see what happens. So what happens in a model that everybody knows about if I interject hands-on healing or distant healing or healing with intention? That's the question. So here's what happens. Short version, it's ugly. This is a mouse at the end of its life, dying as it should. If I don't know if I have a pointer there, but I can see a pointer. I hope everybody can. This is the tumor. It's big. It's ugly. You don't have to be a biologist to figure this out. The fur is falling out. If you could see the eyes, there's nobody home. This mouse is within hours of dying. And what I do is I interject a single variable. And so this is a picture of me at Brown University, because I don't out my students and faculty volunteers who also take part in this. So this is me at Brown University, putting my hands on a cage of mice and then doing my healing technique. Now, incidentally, this is pre-COVID, so I think I was precognitive. So we in order to do this stuff, you got to wear hats and gloves and things, and you got to be scrubbed down. I look normal now in COVID land, but before, this is exotic if conditions were a little bit more normal. So we know what's supposed to happen. It happens everywhere. And we introduce a single variable, and then we look to see what happens. And what happens is, to my surprise, tumors grow. They begin to ulcerate. They really ulcerate. Now look at this mouse. Look at the size of the ulceration. And if we do histology here, we got viable cancer cells, but the thing has split open. The coat is clean. The coat is nice. If you could see the eyes would be clean. And the mouse is running around in the cage. Notice we've already broken the world record of how long you live. And then a couple of months, maybe a month down the road, by a month down the road, this mouse will be completely healed. I don't know if I brought completely healed pictures. It'll be completely healed and live its normal lifespan. In other words, the mouse will be cured. Won't be remitted. It's not a suppression of symptoms. The healing cures the cancer in the mouse. And so the question is, what in heaven's name is going on? and what are the correlates? So if we had time for separate talks, I could defend, I think, with reasonable amounts of data, that healing is reliable. I've done this 18 times. 19 and 20 are just begun. And so healing can reliably happen. And of course, reliably happen is part of what you'd need to in order to be conventional. I think I could defend, if given with sufficient data. Second bullet, there's a dose response. In other words, this isn't magic. It's not, you wave your hands and magical things instantly happen. Nothing like that goes on. There's a real dose response. There's too little treatment. There's too much treatment. Too much treatment doesn't hurt the mouse, but it doesn't help. And so we can start to model what's necessary in order to cure cancer. I could defend, if with data, that healing proceeds non-linearly. And what that means is you don't just start to get better, whether you're a mouse or a man. You don't just start to get better. Healing occurs in sudden bursts. So there's a treatment going on. It seems like nothing's happening. There's a treatment going on. It seems like nothing's happening. There's a treatment going on. It seems like that. And then suddenly things change. I could show you mouse pictures, but we go down a different road where you're looking at an ulcerated mouse like you just saw, and suddenly it's gone. I mean, suddenly it's gone. It's quite startling. I'm reasonably confident, and I think I could defend with data, that there is no energy in energy healing. I mean, I have a book called The Energy Cure. I told the publishers, I don't think there's any energy, and they said, thanks for your input. They didn't care. I think it's information. It's not energy. It's not energy. I mean, look at a simple example. All energy diminishes. You can model it by a very simple formula. If I'm talking and I move farther away, it gets quieter and quieter and quieter. If I shine a light, it gets duller and duller and duller. All energy diminishes with distance. Healing doesn't. Now that's reasonably interesting. You can do an experiment with mice at 2 inches, replicated at 2,000 miles, and you get the same result. What in heaven's name could that be? Because it certainly isn't energy. And everywhere people talk about energy healing, yeah, I don't think so. I could defend, different talk, that there is a bond which occurs between healer and healee that is fluid. And I could show you stuff from EEGs, I could show you stuff from functional MRIs, that in fact, if you're dealing with people, if I've got a healer and a healee, the brains go into a phase lock. I mean, this is seriously detailed. It goes into a phase lock. Something bonds people, and they bond them at a distance. We're about to start, incidentally, to see how much we're actually bonding with mice. This is an experiment about to start. I could reasonably defend this bullet. It's not due to a particular state of mind. You don't have to even be aware that you're healing in order to heal. You don't have to believe in order to heal. You don't have to do anything like that. That healing seems to respond to life in need. And we can look at the brains of volunteer healers. They're not really healers, but the brains of volunteer healers will turn on if presented with a healing problem, if presented with a healing need. And maybe we'll touch on that later, maybe not. And so there's a bunch, yeah.
Sorry, can I break in with a couple of questions?
Break away.
So 2 things come to mind as I list, or I see this list of things that you've discovered over the course of the past 35 plus years. First, we have a question from the audience. Kathy asked the question, and you're talking about this bond between the healer and healee. And she asks, do you feel like anyone can learn energy healing? And then the other question relative to this slide I'd like to ask is, when you say it's fundamentally information, can you give us a sense of what you mean by information?
Yeah, I'm still trying to figure out what information is. So I'll answer the second one by saying, well, information isn't energy. Now, it's possible that information and energy are fungible, but what I actually think is going on is that in an analogous way to, say, a radio station, you have a power source that's energy. There is a power source to the planet. There are planetary waves that I think is really the power source behind this. But if I just have a power source and all I have is a bunch of power, I got to put information on top of that. So if I'm in a radio studio, I start to speak. I'm giving information from when I'm speaking that then rides the power source out. I think that's actually what's going on. I hope that's helpful, but that's as close as I can come to this. In terms of whether anybody can heal or be taught to heal, I think it's still an open question. I wrote a paper once for a journal entitled "Can Healing Be Taught?" And I wrote it with a biologist buddy of mine, and we had done a number of mouse experiments where we got inexperienced volunteer healers, inexperienced volunteer students, inexperienced volunteer faculty to come and learn the method, and then they got mice, cancerous mice. They all cured them. The only caveat I have, can everybody heal? So far, I haven't found anybody who can't, but I don't know if believers can heal. I know that sounds silly, but I'm not trying to be dopey. I don't know if believers can heal because I never work with believers. I myself am a skeptic. Skepticism has gotten a bad rap. There's believers that come in all sorts of flavors. There are people who believe everything is true. There's people who believe everything is false. I've given talks, for example, at skeptic societies, and I start out by saying I'm the only skeptic in the room. Everybody else has their mind made up. You already know that everything I'm going to say is wrong. These aren't skeptics, they're believers. I don't know if someone locked into a belief system can heal. So far, I haven't found anyone who's inexperienced and skeptical who can't. But I don't know, and I can't generalize whether believers can heal. So, for tonight, can we store and reproduce healing? Whether it's produced by a believer or a non-believer, whether there's energy or there's not. If we can produce the phenomenon, can we store it and reproduce it without the healer? So think, I don't know if you can see me in a little box, but think, I mean, this is just a memory stick. Think like a battery. So I'm storing stuff in the battery. It stays in the battery for some length of time, and we can draw upon it to get it out. Now, you don't have to believe in batteries. Batteries work. And similarly, healing, you shouldn't have to believe in healing. I mean, does healing work or doesn't it work? I mean, it's a pretty straightforward question. And so can I store this thing, which I'm alleging works, and reproduce it without the healer? Can I have a whole bunch of these things? In other words, can I make it scalable? And if it can be made scalable, can I turn it into a conventional or a more conventional kind of problem? And looking ahead, and I have experiments going on right now, healing does seem to be able to be stored. I'm going to show you some of that. And it seems to be able to be stored in stuff. It can be stored a variety of ways, and the stuff that it's stored in can be organic and inorganic. It could be either one. And I don't know so far whether there's really a preference of one over the other. But healing, if we're looking at a battery, the question would be, well, what should we make the battery of? How do you engineer this? And we've done it in organic and inorganic materials. And that's the stuff I'm going to show you. So a couple of illustrations. Cotton. I don't have a piece of cotton with me. Cotton, this goes to back to the great Bernard Grad. If anybody is a scholar of the history of healing research, Bernie Grad at McGill University in the 1950s and the 1960s, I think is the granddaddy of them all. I'm in his lineage, I hope, but he's the great Grad. He's no longer with us. But among the things he found is you can store healing intention in cotton and water. And I've taken that off and maybe gone a little bit farther, but that's the general plot. You can store it in cotton and water. And what that would mean if you were looking at it is if I were holding a piece of cotton as opposed to holding a cage of mice, and I did my healing stuff in tension, the cotton takes on, I don't know the right words. It takes on the memory of this. It takes on the information of this so that the cotton can be used as a healing surrogate. So instead of having someone, again, I'm assuming you can see me, put their hands on me, I could take a piece of cotton and put the cotton on me, and I would get, I'm hoping, similar results. And we've done a bunch of tests, lab tests on this, as well as tests with animals and anecdote with humans. And it turns out that cotton, if cotton that's been charged, that's been intended, that's been treated, is brought near cancer cells in a dish, in a well plate, test tube, not really a test tube, but in a well plate, in a bowl, if we have a batch of cancer and we bring charged cotton to it, the cancer cells can recognize that the cotton's been charged, and they genomically change. So as the cotton gets closer to the cancer cells they react, and they genomically change, and proteins fold, and this happens and that happens. And if the cotton has not been treated, you bring it near cancer cells and they, what do you want? Get away from me. They can tell. If you have cancerous mice and you put cotton near the cage, they'll move to the cotton. Once they're cured, they don't go to the cotton. And it turns out, this turns out to be a potentially interesting area. The detector of healing seems to be necessary that the detector be alive and that there is a healing need. If the detector is alive, but there is no healing need, it doesn't react. And I can give you a quick example from bacteria studies and other types of studies with cells that have motility. If we wound cells in a dish in vitro and we bring cotton near it, the cells, if they have motility, will swim towards the cotton. They'll swim towards the cotton until they're cured, then they don't swim towards the cotton anymore. Single-celled organisms can recognize whether cotton has been charged, but I've put cotton under, for example, an electron microscope, looking for structure. Nothing. I can't find anything. I can't find any way so far to have a helometer that's a thing. The helometer, and I'm actually developing this in Providence, the helometer seems to necessarily be alive, and it seems to necessarily have a need. So it's, again, interesting. So if we bring cotton near cancer cells, I'm not going to go through this, they genomically change, and the proteins fold, and this happens, and that happens, and things like that. We've done studies with cell medium, and this is just the off-the-shelf nutrients that you buy and you use in labs, and you dump a bunch of cells into this stuff, and it keeps them alive and keeps them healthy and all of that. So we treat the cell medium, not the cells. And then we grow cells in the treated cell medium. So this is organic. And we pour in a bunch of cancer cells. It's a little more elaborate than that. We pour in a bunch of cancer cells into this medium. And what happens is the cells wildly respond. Now, they don't do it immediately. It has to be that phase transition. It occurs in bursts. First week, nothing happens. Second, this was done at UC San Diego. In the second week, the cells explode in growth. The cancer cells increase by an average, here it is, 600% increase in growth. Now that's not subtle. That's not something you want to do statistics on and see if we can tease something out. This is in your face. And so the cells go crazy. They can tell that the medium is, of course, if the medium isn't treated, they just go along being what they are. But if they've given this charged medium, the healing intention medium, they go nuts and they use it to their advantage.
Bob, or Bill, rather, can I break in with a couple of questions?
Go ahead.
Bob wants to know, does the charged cotton drain like a battery? Does it drain? And if so, does it need to be recharged? Andy is curious as to whether or not any of this research has been published in a cancer-related journal. And then short question, but probably very long answer. Mark is wondering what the healer is actually doing.
That third one I can't do. You know, that's going to have to go to, yeah, that's going to have to go to the published sources. But you know, we'll kill the rest of the time and won't get to the plot line. So I don't mean to duck it, but I'm constrained by the clock. In terms of cancer journals, no. Everything they've been submitted to cancer journals, they have said we don't publish this kind of crazy stuff. At the same time, I've done this in departments of oncology. I've done this in 6 medical schools, and I've done it in the medical schools in the departments of oncology. So I've given, for example, at a large cancer center, I've given a talk, and there were probably 75 oncologists there, and they gave me a hard time and tried to cut my knees off in all sorts of different ways for hours. And at the end, I'm not trying to brag, but I got a standing ovation because they couldn't find a methodological flaw. I said, if I'm doing something wrong, let me know. Show me how I'm screwing up. And at the same time, they didn't know what to do with it. So they said, that's amazing, but what do we do with it? And at the time, I didn't have the practical stuff that I can now apply. So many people know about this. I can tell you with certainty, there are physicians all over the world practicing this quietly because they're afraid to lose their medical license. And I'm not a conspiracy theorist, but they're afraid to lose their medical license. I was supposed to be giving in April, for example, a lecture to a group of medical doctors in the Netherlands. We were expecting something like 150 medical doctors who wanted to do this themselves. We got COVIDed, so I couldn't get there live, and this happened and that happened. COVID happens. I'm giving a lecture to a group of physicians in Germany at a medical conference coming up, I don't remember, end of March, early April, something like that. But it's not a secret. There are physicians all over the world, and I get anecdote from them going, they just cured, they just cured, they just cured, they just cured. They can't say it out loud. And depending in the States, it depends what state you're in, whether you're allowed to do this. So I've given workshops to physicians. I've given workshops to veterinarians. Veterinarians can cure dogs and cats, everybody's happy. But if you cure a person, be careful. Be careful. And there are rules against that kind of stuff. So it depends on the state. I don't know about Colorado, but New York State, you got to be careful. California, to my surprise, you got to be careful. The state which is the most lenient and doesn't get upset if people get cured, I would say, is probably Arizona. In Arizona, people can be cured the same way that people are happy if you cure a dog. you cure a person in the wrong state, you're screwed. I've taught groups of people in Ontario, in Quebec, and physicians only, and they all say, we have to sneak around when we do this. So the answer, it's not a secret. This would be known in many, many parts, but I can't, so far I've gotten into conventional biology journals, but I haven't gotten this into one explicitly devoted to cancer.
Well, I just wanted to tag on to Bob's question about the cotton needed to be charged. Damon has also asked if there's something specific about the nature of cotton for holding information.
I'm working on that. So far, I can't figure out what it is. This is a total ripoff off the great Bernie Grad. It turns out once cotton is used, it seems to take on the memory of the thing it treated and does not seem to be able to be recharged or reused. So if you've used this to treat a torn ligament, then you don't want to use it anymore. You almost treat it like medical waste. If you've done this to fix yourself of a leukemia or of a tumor, you don't want to. It doesn't seem to be able to be recharged. And this is another area of inquiry that I'm trying to go down and figure out what in heaven's name is this, and what are the patterns of storability. So all good questions. Here's mice. Now, this is just ripped off the internet, you know, pictures of mice. This isn't a mouse, it's a rat, you know, but we're dealing with mice, but as close as I could find. On the left, we have a mouse that was treated using the healing technique. And on the right, a mouse that has not been treated using the healing technique. So this mouse has been treated, has gotten ulcerated, is on its way to cure. And we take blood or we take cells from this mouse and we put it into this mouse and everybody's cured. The mouse on the right has not been treated by a healer. And so we've taken the information, and I'm using information deliberately, on the mouse on the left, put it into the mouse on the right, and it's cured. Now you can see the analogy here to, dare I say, making a vaccine. I suspect that we have enough information now that we could make a cancer vaccine. Needs to be tested, should not believe me, but it seems to be the case. We don't have any indicators that this is not a transferable piece of learned immunity. If we switch over to inorganic material, for example, water, we've done experiments with water bottles filled with water that we treat and we feed the water to mice that we never meet. And all of those mice are cured. And so something's going on in the water. So the same question that I was asked before, what's going on in the cotton? I don't know. What's going on in the water? I don't know. I have a series of experiments on the drawing board, but once again, they got COVIDed and sidetracked, but we'll be working at the University of Washington, hopefully when COVID is over, and be actually looking into a deep dive into what in heaven's name is happening to water. We know, for example, that the bond angle of the water changes, the angle of the hydrogen to the oxygen atoms, but we don't know why that's relevant, or we don't know enough. So we can store this stuff in organic material, we can store this stuff in inorganic material. Let's abstract a little bit now, and can we simply record what's going on and reproduce this with a recording? So this is my team to work on that. As president of the SSE, I have some really good folk to draw upon. And so I exploit people all over the place. I call these my partners in crime. On the left, you probably don't recognize, this is John McMichael. He's a virologist, an immunologist, and probably a bunch of other ologists. And he is just extraordinary. Boy, you want to get into how somebody could revolutionize medicine. It's the guy on the left, John McMichael. Many of you may recognize, if you're interested in consciousness stuff and all that, Dean Radin is the second one in. And so Dean is happily joined the team, and we've been working on, can we record, can we store, can we do all that? Third guy in is a guy by the name of Mike Turner. He has a PhD in signal analysis. There's not a lot of people who have PhDs in signal analysis. And Sarah Besum on the right does the day-to-day stuff in the lab. And what we did here is we got the team together, designed a reasonably interesting study, and we carried out the study inside of a Faraday cage. This is a big walking Faraday cage, 12 feet by 12 feet. And inside of a Faraday cage, you have, it's shielded from most electromagnetic contaminants. All of us now live in a very polluted environment of electromagnetic craziness. And if you're doing recordings and magnetic things and electronic things, you need it to be a safe place. And I got to tell you, this is just an endorsement. If you ever get a chance to get inside of a Faraday cage, take it. And then you'll understand the crap you're living in. We've upped the electromagnetic pollution step by step by step by step by step by step by step until we actually consider what's going on to be normal. And if you ever get into one of these places that doesn't have the pollution, you're going to go, oh my God, this is what it's supposed to be. And you don't have to be particularly sensitive. It's really quite astonishing. So anyway, it's a big wonkin' thing. And inside of it, we put all sorts of stuff and all sorts of detectors. And we since we don't know what we're looking for, we just kind of throw the kitchen sink at it. And we put detector upon detector upon detector even caduceus antenna, and we're doing scalar field detectors, and we're just what can we get, and let's do the whole thing. So we throw the kitchen sink at the problem, and we put a bunch of stuff in there, and we run it through a water-filled supercomputer that was made for us, this monstrous 38 different kinds of detectors into a recording. And the recording, since we're kind of making it up, kitchen sink, the recording has all of the detectors converted to an audio file. It's a very thick audio file. Then we take this recording and we play it to cancer cells, and we play it to cancerous mice. So what you're looking at here is an incubator. It's in Providence. And we got a regular amplifier, except it's probably not as good as the one you have in your house. We were cheap. We spent a lot of money on setting this thing up, way into the six figures. And now we have a $49 amplifier, which we got from Amazon. And we have $49 speakers, which we got from Amazon because we knew they would fit into the incubator. That was our rationale. So we got this zillion-dollar recording, $49 speakers, and $49 amplifier. And down here are cancer cells. And so there's a computer someplace on the side, and it's playing this recording to cancer cells. And we try dose responses and all those kinds of good things. And we play this to, this is the geeky part of the program. We play this to cancer cells, and we do all sorts of genomic studies and checking out this and checking out that. And the real short version is this. We looked at 167 genes connected to cancer, and we play the recording, and 68 out of the 167 genes change when we play the recording. This isn't subtle. And it changes reliably. And we did 7 iterations of this thing. And when the recording is played, the cancer genes go fold, 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 do this, do this, do this, do that. And we're starting to figure out what the most promising ones are. I'll give you just a quick look ahead. One of the things that happens is that apoptosis is upregulated in cancer cells. Now, this is big. Cancer cells are immortal. Cancer cells are perfect. Cancer cells have no off switch, and they like to grow. Normal cells, wherever you got them on you, inside or out, have a lifespan, and then they self-destruct, and this is called apoptosis. Cancer cells don't have apoptosis. They just keep growing. They don't know when to stop. When we play the recording, they start to stop. That's reasonably interesting. So there's stuff that happens in the immune system. There's stuff that happens to this. There's something that happens to telomere length, a lot of this stuff. And this goes to your question that I got earlier that Claire asked from somebody else. Have we gotten this into a cancer journal? No, but this is dose response. This is a traditional biology journal. And they couldn't say no. I mean, where did we make a mistake? So the partners in crime, we put together, I think, a reasonably interesting paper that is published in a conventional biology journal. And our conclusions were that we can do this reliably. And it's reliable, and we get 68 out of 167 cancer genes affected. Number 2, it's a proof of concept. We can capture and release healing intention. Number 3, this is an interesting finding. The dose response, and I'm ducking how we did this, but the dose response, the strength of it, let's call it the strength of the recording doesn't seem to be as strong as doing hands-on, like we put your hands around a cage of mice. You put your hands around a cage of mice, and stuff happens at a particular time and place and all that. You put your hands around a cage of cancer cells, and something happens. The recording reproduces a bunch of it, but it doesn't seem to be quite as strong as live, and we don't know why. So the possibilities are live healing is, you could call it analog, and this recording is digital. and digitization of anything reduces information. So it's a sampling technique, even though it's very dense, it's a sampling technique, and we may have lost information by the digitization process. It's possible we don't have the right detectors. It's possible we're not asking the right questions, but it's also possible that this recording eventually will reproduce what we can do live. If we can reproduce what we can do live, now think of the scalability potential. So let's say, just a thought experiment, I'm not claiming this yet. Let's say the recording reproduces the cancer cure. We can upload the recording into the cloud. It becomes, you talk about scalable, this becomes instantly global. That's reasonably interesting. We only published the first 3 conclusions because the 4th we thought was too much. And so I can tell you I can defend this, but I didn't put it into a peer-reviewed publication. And that is that when you play the recording, you produce a conditioned space in the room. So you saw before in terms of the incubator, we can play this and we get the genomic changes, but even after we turn it off, 3 days later, we've stuck cancer cells in there and they still genomically change. Something happens to the space when this stuff is played. And we have many examples where this has been detected. It's very, very interesting. If anybody has been exposed to, for example, the work of Bill Tiller on conditioned space, ours seems to be going along in that line also. So we've done this also with mice, and we did this, this was done at Brown University, and we exposed the mice to the recording. This is the exact same stuff you just saw. These are the same speakers that were inside the incubator. This is the same amplifier, but now instead of cancer cells in an incubator, we have cages filled with mice. So the question is, what happens when you play this recording to caged mice? And here's what happens. We started to get the same kind of ulcerations. This is just close-up of some ulcerations in some of the mice. Some of it's a little bit ugly. But what happened in this case, when the Brown University Animal Ethics Committee, the IACUCs, saw these ulcerations, even though we told them about them and showed pictures of them, they freaked out and they came in and they killed the mice. The mice were doing fine. The mice were under no stress. The mice were happy. They were eating, they were drinking, but they had this ugly wonkin tumor on the side, just as they do when you do it live. And the ethics folks came in, freaked out, and whacked the mice. And the only way to say it is they whacked the mice. This was a serious setback that we had. Before they were whacked, or actually right after they were whacked, we got some blood, we got some tissue, and we got all that kind of stuff from them. And we turned this into also a very good paper. It's a very good paper, if I do say so, in a traditional biology journal. And what we found, it was working. We don't know if it would take the mice to full cure, but we had changes in all sorts of stuff. So I'm not going to go geeky on you, but there were, just look at the last one. Spleen weight, this is the weight of the spleens of the mice, increased 5-fold. So we're not talking little things, we're not talking statistical effects. We're talking 5-fold. An increase in spleen weight is associated with a stimulation of the immune system. So would the mice have gotten cured? I don't know. And I've spent the last more than a year looking for a lab that'll carry this out all the way to the end and not let the animal ethics folks, if the mice are otherwise healthy, not let the animal ethics folks whack the mice. If the mice are suffering, you whack the mice, no problem. But if the mice are happy and playful and hanging out, you don't whack the mice. So I just started an experiment in a lab, I won't tell you it's Tokyo, where they have agreed that we're going to carry this out and see what happens and see if in fact we have a cure. If we have a cure, that'd be reasonably interesting. But for our purposes here, it would become instantly scalable. I gotta try to speed this up. I got water, and I'll just say this cut through. I already told you that we've done experiments with treated water, treated with hands-on, fed to cancerous mice, cures those mice. But can now we make this mass producible? Same thing with cotton. Cotton may work, but how do you get cotton to be treated? Can we scale that up? And so my question is the same here. Is there any way to enhance treated water? And what we did is we did some dilution and succussion, and try to wrap yourself around diluting water with water. And we settled on, we did a lot of these things. And we started to come up with a dilution and succussed treated water, which can be mass produced. And we did a pilot study of about 100 volunteers, and they took 2 drops 4 times a day sublingually for 8 weeks. So again, we've gone from cells to mice to people. And we found, to our utter astonishment, and frankly, skeptical astonishment, 98% reported improvements in health. All across the board, physical changes, mental changes, what we call spiritual changes. And the stuff that people were reporting was astonishing. They were talking about, we'll just say, effects on their cancer, effects on their arthritis, effects on their depression, their anxiety, their pain, their Parkinson's. Astonishing array of things that this juiced up, we'll call it juiced up water, seems to be able to affect. Now, the effect was so interesting that we right away replicated it. We replicated it to about 100 volunteers in Europe, and we got essentially the same thing. And being real thick and a little slow on the uptake, I said we got to now see if we can mass produce this water. And so we did a system where actually putting water through a physical device that we invented to mass produce it, and we gave that water to a 3rd clinical trial of 100 people. So we have 3 clinical trials having the water produced using different systems, and we got essentially the same thing. So we've got now roughly 300 people, it's actually higher, but roughly 300 people who are just saying, this is only anecdote. So they're not in little cages and we're not controlling their environment, but they're saying, I need more water. People do not want to give this thing up when they start it. Now, one of the interesting things is that some of the effects appear to be stronger with the water than with the live healing. Now, this is in contrast to the recording. The recording may get to the same end, but it takes a longer period of time. It doesn't seem to be as strong. The water seems to be stronger. This is quite mysterious to me. So we've had, for example, very little effect on Parkinson's patients using hands-on healing. Just not good at it. But in the case of the water, there's all these anecdotes that come at us that are confusing. We've had people in wheelchairs give up their wheelchairs. I mean, that kind of crazy stuff. For some reason, the recording seems to be perhaps a little weaker. The water seems to be a little bit stronger. So what can we do with the recording and what can we do with the water? And now we're going, I don't know. What happens if we condition the space of a treatment room? What happens if we supply people with treated water in the midst of whatever therapy they're doing? What happens if we play a recording? What happens if we combine a recording and water? I mean, you can see it's almost endless, but we're going endless with only those things which are scalable. So I'm watching a clock here. Special thanks to Brown University, Institute of Noetic Sciences, Beech Tree Labs, and incredibly generous funding from Emerald Gate Charitable Trust. So I'll take any questions in the remaining few minutes. Thank you.
Thanks, Bill. Yes, we just have a few minutes and I'm hoping we can do this as a little bit of a lightning round. Anne has asked a question, which I think you may have sort of just addressed, which was, have you tested this on anything other than mice with cancer. And it sounds like the water experiment was in humans.
Yes, three different clinical trials in humans, and it seems to be really robust.
Mark is wondering, relative to that experiment, was there a control group?
No, not in the clinical stuff yet. We're doing a proof of concept, and we had sufficient anecdotes to start that in order to get the volunteers, we told them they're not getting placebos. So we didn't say, you got to, I understand the placebo controlled and all that. First, we wanted to see what happens, to see if it was worth it. And if it worked, frankly, we didn't want anybody to get, these were people in need. These weren't just 100 people off the street. We recruited through various networks people with serious conditions, and so if you've got a serious life-threatening condition, I'm not going to at this point, with the level of information I have, put anyone through a placebo-controlled trial.
So Ken has asked whether cancer is the only disease this technique can be used with, and I'm just also wondering, along with that, if you would address what this might be effective against and what it's perhaps ineffective against.
Yeah, as a generic rule, and rule is too strong, we're better at taking away things that you don't want than in stimulating things that you're missing. So, for example, it's cancer, you got a tumor, you don't want the tumor. Alzheimer's, we have startling results in Alzheimer's. I mean, it's just simply, I don't believe it. But on Alzheimer's, you got plaque on the brain. The plaque goes away. We've done inflammation studies at a medical school. We're astonishingly good at inflammation. So anything related to inflammation, we're good with allergies, we're good with colitis, we're good with diverticulitis, we're good with mental disorders of depression and anxiety and bipolar. We got people off their meds. We got all that kind of good stuff. We're not so good with, I don't know of a single cure of type 1 diabetes. And that's an example of I'm missing something. With the water, we're getting results in Parkinson's we didn't get before, but it's too early to plant the flag on that, because in Parkinson's too, you're missing something. In diabetes, you're missing something. In Alzheimer's, you got something you don't want. And that would be as kind of a working hypothesis. But even there's some exceptions and some things that simply, almost impossible to make sense. We drastically accelerate the rate of healing. So if you were just injured, if you have a torn meniscus, that's going to heal very quick. If you have a ripped Achilles tendon, that's going to heal very quick. So you have a general enhancement of healing, but it's not magic, and it doesn't fix everything. And there's some stuff we've just had outright failures. Diabetes, the most extreme diabetic for type 1 that I know of in the many people that have been tried, we had a guy who had all sorts of body parts missing, because he had it too long, too severe. He was completely blind. We gave him about 50% of his sight back and about 50% reduction in insulin. He was happy, but it didn't go. I couldn't get it past that. So I couldn't fix it. And it's not just me, other people, they couldn't fix this. So we haven't cracked the code really yet, code being too strong, of how do you stimulate stuff that you're missing? We're not particularly good at it.
Okay, so Ken has gone over to the dark side and wants to know, is there a danger this technique could be weaponized to cause disease?
I don't think so. I don't think so. You'd be in trouble, because I think when you give a treatment, you get one. So I think it would have to be a self-destructive person that would try to do that. And I would be happy, because I'm a data freak, to train people to try it. But I'm not doing it, because I see too much of this stuff comes back at you.
So we really are out of time, but I'd like to give you the last word, Bill. What would be the key takeaway for our audience tonight?
This is a seriously interesting and scientifically explorable problem. And the idea that you walk around and say, I wonder if healing is real, is crazy. It's crazy. There's too much data. Now that doesn't mean every person who claims they can do whatever they can do it. And I don't have anything good with comparative healing techniques. I just research my own, and so I'm a little bit too much inbred. So I'm this is not an endorsement of anything. Certainly not other healing techniques. I don't know what they can do and not do. There's so many interesting questions to do, but this is not about belief. It's not about spirituality. It's not about, gravity is interesting too.
Bill, thank you so much for this fascinating conversation. Everyone at home, thank you for joining us. And we have another fascinating conversation coming up a week from tonight. We'll be discussing Germany after the end of Angela Merkel's tenure as chancellor with 2 German experts. Thanks, everyone, and have a great night.
Thanks.